Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. My name is Iqra, student of Chemical Zoology, Department of Zoology, Kohar, University of Science and Technology. The topic of my presentation is effect of neem leaf supplemented diets on growth and hematological parameters of grass carp. Presentation outline, introduction, objectives, material and methods, results, conclusion, recommendation, acknowledgement, references. Introduction. The neem tree, botanically referred to as Azadirachta indica, belongs to the family Cyprinidae. It is a fast-growing evergreen tree that may grow up to 20 meters in height. It is one of the most useful traditional medicinal plants. Almost every part of the neem tree, including seeds, leaves, roots, bark, trunk, and branches, have multiple uses, but most of the pharmacological activities have been described especially for neem leaves. This species, which I was used and now researched, is grass carp. Grass carp is a herbivore. Grass carp scientific name is Stenocerandodon idela. Grass carp is one of the essential freshwater aquaculture species with the top production among all species around the world. Growth in grass carp is a function of age, size, abiotic factors such as density, nutrition, temperature, and oxygen. Grass carp requires cellulose and protein in their diet, with protein being especially important for optimal growth and young fishes. Objective. The first one objective is to evaluate the potential effect of neem leaf supplemented diet on growth performance in grass carp, and the second one objective is to determine the effect of neem leaf supplemented diet on hematological parameters of grass carp. Materials and methods. The first one is sample collection. The second one is leaf collection. The third one is diet preparation. The fourth one is growth analysis. And the fifth one is hematological analysis. And the sixth one is statistical analysis data. Here we are seeing two tables of result. The first one table shows growth of each fish and the aquarium before supplemented diet, before exposed to supplemented diet. The second one table shows growth of each fish after exposed to supplemented diet. Here we see clear difference between these two tables, but for further clarification, I have got two graphs for these two tables. The first one graph is for the first one table, and the second one graph is for the second one table. And we saw positive effect of supplemented diet on the growth of fishes in these two graphs. This one is the second graph for second table. Here we can see clear difference between the growth. The concentration of neem in the experimental diet is the 2 gram in T1 and 5 gram in T2 and 10 gram in T3. Results. Hematological analysis. Red blood cells, lymphocytes, eosinophils, monocytes, basophils. These are all the hematological parameters. First one is the graphical representation of hematological parameters. Red blood cells, red blood cells. Red blood cell count of blood of grass carp species decreases as the neem concentration in the feed increases. The second graph shows that the lymphocytes count of the blood in grass carp increases as the neem concentration in the feed of fishes increases. The third one is hemoglobin parameters, hematological parameters. Hemoglobin counts of blood in the grass card is decreases as the mean concentration in the feed increases. Similarly, the fourth one, eosinophil. Eosinophil and monocytes. These two are similar as the mean concentration in the feed of the fishes increases. These both parameters are also increases. I didn't draw graph for the basophils because the mean concentration, mean diet have no effect on the basophils. Conclusion: neem leaf supplemented diet are more effective compared to normal commercial diet of fish. The second one is neem leaf supplemented diet acts as a growth or immune enhancer. The diet prepared from neem leaf are cheapest as compared to commercial fish diet. Neem leaf inclusion in feed has a remarkable positive effect in the improved general growth performance. Recommendation: neem leaf supplemented diet of fish should be prepared with high concentration of neem. Its negative effect at high concentration level should be recorded. Improper technology should be standardized to alleviate these effects. Management, I am very thankful to my supervisor, Dr. Farmanullah Davar, and my co-supervisor, Mr. Salman Ahmed for their guidance and support. Thanks to all faculty members, my friends, my family, and also my lab fellows. These are references. Thank you for your attention.